Welcome to The Explainer. Today, we're basically going to be detectives, embarking on a quest to uncover the secrets of life itself. We are going to crack the code that makes every single living thing on this planet what it is. I mean, really think about that for a second. A flower, a whale, you and me, we all start out as this tiny single cell. So, what kind of instruction manual, what molecule could possibly hold the blueprint for everything? From a petal to a fin to a human brain. That was one of the biggest mysteries in science. So our story really kicks off with this massive hunt for the physical stuff of inheritance. Scientists knew that whatever this mystery molecule was, it had to be amazing at two things. First, storing a colossal amount of information, and second, copying itself perfectly over and over again. The search was on. Now, this hunt took decades. It all started with a clue back in 1928, when a scientist named Griffith noticed some kind of transforming principle could magically turn harmless bacteria into deadly ones. By 1944, a team of researchers had a prime suspect, a molecule called DNA. But, most of the scientific world was pretty skeptical. They thought, no way, DNA is too simple. They bet all their money on proteins, which are way more complex. It was going to take one last, absolutely brilliant experiment to finally settle the score. Right, so this was the heavyweight championship match of biology in the early 50s. Protein versus DNA. To figure it out, Alfred Hershey and Martha Chase came up with this incredibly elegant experiment in 1952. They used a virus that infects bacteria. It basically works like a tiny hypodermic needle, injecting only its genetic material inside. Their goal? To figure out if that injected material was protein or DNA. Here's the genius part. They knew that proteins have sulfur, but no phosphorus, and DNA has phosphorus, but no sulfur. So they could use this to tag each suspect. They took one batch of viruses and labeled their protein coats with radioactive sulfur. For another batch, they labeled the DNA inside with radioactive phosphorus. Then, they let the viruses do their thing, infect the bacteria, and then, get this, they put them in a blender. Seriously, a kitchen blender, to shake the virus shells off the outside of the bacteria. The result was just undeniable. The radioactive sulfur, the protein, stayed outside, but the radioactive phosphorus, the DNA, was found inside the bacteria, hijacking the cell. And just like that, it was game over. DNA was the one, the molecule of heredity. But you know how science is? Solving one massive mystery just ripped the lid off an even bigger one. How in the world does this molecule actually work? Okay, so with DNA finally crowned the king of genetics, the next great race was on. Figure out its 3D structure. Everyone knew that the shape, the actual architecture of the molecule, had to be the key to understanding how it could possibly store and copy the entire encyclopedia of life. A huge, crucial piece of this puzzle came from a chemist named Erwin Chargaff. He was just painstakingly analyzing the chemical makeup of DNA from all kinds of creatures. And as he crunched the numbers, he found this really bizarre, really consistent pattern in its 4 chemical building blocks, or bases: adenine, thymine, guanine, and cytosine. What he found became known as Chargaff's rule, and it was deceptively simple. The amount of A in any sample of DNA was always the same as the amount of T, and the amount of G was always the same as C. It was this weird little coincidence that nobody could explain. At least, not yet. And just look at this data. This is what makes it so cool. Doesn't matter if it's a virus, a bacteria, or a human being, the pattern holds. The percentages of A and T are almost identical. Same for G and C. This wasn't just random chance. This was a massive clue, screaming that these bases had to be pairing up in some way. And that pairing rule, well, that was the breakthrough. You combine that with the absolutely critical X-ray images from Rosalind Franklin, which showed the molecule was a helix, and suddenly James Watson and Francis Crick had everything they needed. In 1953, they put it all together and proposed the iconic structure we all recognize today, the double helix. And the structure itself is just brilliantly simple. It's like a twisted ladder. The sides are a sugar phosphate backbone, and the rungs? Well, that's where Chargaff's rule comes in. A always pairs with T, and G always pairs with C. These pairs are held together by hydrogen bonds, strong enough to hold the code together, but weak enough to be unzipped like a zipper, which is perfect for making copies. The structure literally explained its function. It was amazing. So we've got this incredibly stable code, this beautiful way to store information. But life isn't just about being stable, right? It's about diversity, variety. So how do you get all that variation? Well, it turns out the genetic deck isn't just copied. It's constantly being shuffled. Even something as simple as a bacterium has really clever ways to mix things up. They can slurp up useful bits of DNA just floating around. They can build a tiny bridge to a neighbor and directly hand over some genes. Or they can even get a special delivery of new DNA from a virus. This constant trading is a huge reason why bacteria can adapt so fast, developing things like antibiotic resistance. But that brings up a really interesting problem for more complex life. Think of a chromosome like a string of pearls, with each pearl being a gene. If two genes are on the same string, they're linked, right? They should travel together. But we know from observation that they don't always. So how do you shuffle pearls that are physically stuck on the same string? The answer is this absolutely beautiful biological dance called crossing over. When sperm and egg cells are being made, the paired-up chromosomes can actually wrap around each other and physically swap pieces. It's incredible. This process creates totally new combinations of alleles. Those are just different versions of a gene that simply didn't exist in either parent. And what's really amazing is that we can measure how often this happens. Geneticists can do a breeding experiment, and then they count the kids. They see how many have the original parental combination of traits and how many have a new, recombined combo. That ratio gives them a number we call the recombination frequency. And here is the truly brilliant part. That frequency number isn't random. It's a direct measurement of the physical distance between two genes on a chromosome. Think about it. If two genes are really far apart, there's a ton of room for a crossover to happen between them. But if they're right next door to each other, a crossover happening in that tiny space is way less likely. So by calculating these frequencies, scientists could figure out the order of genes and literally draw the first maps of our chromosomes. Now, this whole genetic system is incredibly elegant. But let's be real, it's not perfect. It's built on a set of rules, but sometimes those rules get bent, or broken, or even totally rewritten. And that can have some really fascinating and sometimes very serious consequences. First off, the simple dominant recessive thing we all learned in high school, it's not the whole story. Sometimes you inherit two different alleles, and you get a blend, like a purple and white eggplant making a violet one. That's incomplete dominance. Other times, like with AB blood type, both alleles just show up at the same time. That's called codominance. It's more complex than you'd think. And then it gets even more complex with something called epistasis. This is when one gene can completely boss another one around and mask its effects. The classic example is in Labrador retrievers. One gene decides if the coat pigment will be black or brown, but a totally separate gene acts like a master on-off switch, deciding if any pigment gets put into the fur at all. If that switch is off, it doesn't matter what the first gene says, you get a yellow lab. But the most dramatic errors of all happen at the level of whole chromosomes. This is called aneuploidy, having the wrong number of chromosomes. It's usually caused by a little glitch during cell division called non-disjunction, where the chromosomes just don't separate properly. It's like a printing error where entire chapters of the instruction manual are either missing or duplicated. In us humans, these kinds of mistakes can lead to genetic conditions like Down syndrome, Turner syndrome, or Klinefelter syndrome. It's just a powerful reminder that for all the unbelievable complexity of life, a huge part of normal development comes down to something as basic as having the exact right number of chromosomes. So, we've gone on this whole journey, from hunting for a single molecule to cracking its elegant code, and then seeing how that code is shuffled, changed, and even broken. Which brings us to this final thought. The genetic code is a story of incredible stability, a script copied almost perfectly for billions of years. But it's the mistakes, the shuffling, the variations, the mutations that provide the raw material for all of evolution. The system's perfection is, in a weird way, found in its imperfection. And that beautiful balance between stability and error, that isn't just a footnote to the story of life. That is the story of life.